Thank you. Thanks for the invitation to be here. It's a very exciting conference, and that was a fantastic presentation. Thank you, Dr. Green, for enlightening me and many of us to the work you're doing. The road to getting to targets and getting to a concept for a new therapy is all about what we just heard. What I'm going to talk about today is actually how we're racing forward to bring new therapies to the clinic and to patients so that we can have an impact on unmet medical need. And the title of my talk is Unlocking the Power of Machine Learning and Data at Scale to Deliver with Speed the Best Therapeutic Candidates. And we truly believe and have seen with our own experience that AI, ML, combined with data at scale can have a huge impact on the progress of our therapeutic discovery programs. And I want to start with a bit of a picture of how we think about new therapeutic discovery at J&J. And so I represent the molecular discovery engine of the company. We cover a wide range of therapeutic areas and address many different unmet medical needs in neuroscience, immunology, and in oncology. And the vision that we've put in place for how we're going to tackle the existing unmet medical need is through a modality agnostic approach to R&D. And that is based on our vision and driven by our vision that all therapeutic concepts should be achievable. We have the technology and we have the talent to really make this possible, and that any target that we can modulate to have an impact on disease should be druggable. And so based on this vision, we've put in place a comprehensive modality tool set that we need to address and deliver on this vision. So just a little bit about that in this next slide. We have invested for many years in core modalities, both in small molecule and in antibody therapeutics. We have many very important products on the market, and we have a fully established mature discovery and development engine. We've gone on in the past, say, half a decade to decade to build capabilities in RNA-based therapies, for example, siRNA, in gene therapies, and in immune cell therapies such as CAR-T. And we believe that this set of modalities is the basic palette that we need to modulate biology and to address disease concepts that are coming from our therapeutic area discovery initiatives. And in each of these areas, for example, small molecules and in antibodies, the modalities are getting more complicated. We're trying to do more things with small molecules and antibodies that we've done before. And if we look at siRNA, we need to deliver it to certain places. Gene therapy, we need to do the same thing. And in CAR-T, the discovery and especially the manufacturing of these modalities is very, very complicated. And so there's no doubt that to achieve our mission, we need to somehow combine and leverage strengths across the different modalities. And AI and data at scale is a key component of allowing us to leverage across the different modalities to get this done more efficiently. We do not have unlimited resources. We still operate within the confines of what we can pay for within even a very big company like J&J. And so in order to be successful with our mission and to really accomplish what we want to with such complex modalities, we need to leverage across the platforms. In addition, we all know, and we've seen it many times throughout the week, that the costs of drug discovery and development are getting higher, the timelines are longer, and there's a lot of attrition. Additionally, we're working on harder and harder targets. We're trying to modulate biology in ways that are more complicated than ever before. And no doubt the competition is fierce. So we all want to be there with the best medicine for patients. And so combining all of these challenges, we have to take advantage of the data that we're producing, and AI/ML can really help address these issues. Again, as we all know, there's a lot of hype around this as well. We see it in the press every day. And our mission in our journey within J&J, and I know it's the same with many of my colleagues at other companies, is to get through the hype, deliver on the promise, and show real benefits in our portfolio and bringing new potential medicines to patients faster. And a key component of how we're doing this and how we're delivering this in a meaningful way is to do two things. One is to invest in technology, which I'll talk about in my presentation, and also in implementation, both the technology and how we bring it to life in our drug discovery pipeline and portfolio are equally important to unlock potential. So just a little bit about our journey in AI/ML. So if you go back into the early 2000s, of course, we've always had interest in computational techniques to help with drug discovery. And at this time, back in early 2000s, structure-based drug design and informatics and bioinformatics, cheminformatics were important tools that were emerging. But it was a bit of a niche science, sort of connected only to certain parts of drug discovery. And around, say, the early teens, bioinformatics scientists at J&J started to think about actually what is the broader potential for data at scale, and saw some of the trends that were happening or some of the bits and pieces that were showing up externally about developing predictive models that could be applied to specifically medicinal chemistry. And began then to invest in some of the basics of getting data harmonized and usable to generate models, and created global models that could be then used to predict certain properties, and began piloting in a few different programs. Now, within the next 5 years, things changed dramatically. We went from having global models that were being used a little bit here and there on a few programs to actually having new, more sophisticated methods, including generative methods in both protein and small molecule discovery, looking at multimodal, and beginning to apply that to all the programs across a pretty complex and extensive portfolio of programs. And now within the next year or two, our ambition is to take what we're doing from discovery, driving the design, make, test cycle that's largely in the hands of wet lab scientists, and bring this into the automation space, connecting both AI and automation, and having intelligent lab automation drive a lot of the process. And so I think the point of this slide is really to show that it took a while for us to get there, but now we're in a phase of rapid expansion and rapid change. And a little bit about some of the technology innovations. I want to go through a few of the different challenges that we faced and some of the solutions that were put in place over the last, say, 5 to 6 years. One of the big issues that we encounter is if we want to create a tool that's useful in discovery, sometimes that means actually being able to merge data and bring two different very large types of very large volumes of data together in a model that can actually use both the scales of data. And a good example of that is predicting PK, predicting the pharmacokinetics of compounds and how that might perform in animals, how that might perform in humans. We needed to combine lots and lots of in vitro data for ADME properties with a very small set of very different data looking at compound PK. And so we had to explore a lot of different machine learning, deep learning architectures to sort of distill and combine these types of different volumes of data to make a meaningful and useful tool for our scientists. We also encounter different types of data that we'd like to bring together. We've talked a lot about multi-omics. It's been a very big topic at the meeting. We want to bring together structural-based information, 3D information. We want to bring together images of compound activity on cells, and many more different types of data in the same model to actually help us have better predictive tools. And so we have really spent a lot of time exploring model architectures that allow us to bring this type of really pretty complex multimodal data together and have meaningful results again in the projects. Reliability of data, this is a huge factor, especially when trying to convince a chemist, for example, to not make a molecule that they feel in their gut actually would be a good one to make, and to spend that money on a different molecule. We need to know that the models are reliable. We need to show that, and we need to keep track of how our models are performing over time. So we've had to invest in platforms for monitoring and dashboarding our models and to make these accessible to discovery scientists using the models in a real-time basis. And then finally, another challenge that we've encountered is keeping up with a very dramatic, rapidly changing and dynamic portfolio. Of course, as I mentioned, we work across many different therapeutic areas, and the targets and the different kinds of projects that we're working on are very, very diverse. And in sort of the older days, our models weren't always able to keep up with changes in chemistry or changes in sort of molecular features, and we had to refresh and retrain and redeploy models on a much faster scale to keep up. We've actually gone beyond that to really look at new types of model architecture that do not have this same level of erosion. And I'll talk about this a little bit later in the talk. So these technology technology investments, they're actually all over, addressing many different challenges, but very systematic attention to solving the technical problems has been essential, but bringing those to life within the portfolio and implementing the technology has been just as important. And so I want to talk about a couple of examples along the discovery pipeline where some of our tools have been quite instrumental in moving discovery forward. So one platform that we're excited about and that we've been using for the last couple of years in the sort of early hit identification or hit to lead stage of discovery is a platform that we call Sentinel. And Sentinel is really an early warning system, providing us some indication that the road that we're going down might actually be a little bit problematic. Now, the interesting thing with Sentinel is that it actually happens in the background. So if you know a medicinal chemistry project team, there's often not a lot of excitement to find out super early that your lead series or your compounds that you're working on have issues. And so Sentinel works in the background by profiling all the compounds that are either wanting to be designed or being designed and made, and beginning to score them and stack up a score for their risk profile. And as you reach a certain threshold of red bars shown here in the slide, there's a threshold that triggers an automatic retrieval of a compound from our inventory. It's sent out for the relevant assays. It comes back, and if the assays confirm the predictions, there's an alert that's sent back to the team that, hey, there are issues been identified, and perhaps you want to make a different decision or change the way that you're doing your work. And that leads to one of two things. One is you either abandon the compounds in the series, or you begin to deploy more serious models earlier in the process to start to mitigate risk early, or understand if you can mitigate risk early. And so this, I think the key here is that the implementation and the uptake of something that people don't maybe really want to know that early actually becomes operational, because it sort of happens in the background and then gives people some real advice at the time that they want it. And here's an example where we had identified hERG risk in a project in neuroscience. And as you can see, in the red and green bars, the early in the project, before Sentinel had sort of done its thing, more than half the compounds were being synthesized with a risk of hERG inhibition, which is something we don't want. And as the information became known, the project team shifted the way they were doing the work. Models became better, focused on hERG, and most of the compounds being synthesized after this were in the green. And this actually led to more quickly resolving an issue. It also leads to much better use of resources, because of the compounds that we're not wasting money on compounds that are not going to go anywhere. We're making compounds that have a chance of moving forward. So moving on to the second phase of say, small molecule drug discovery is lead optimization. We have many, many models, global models and local models, that we apply to lead optimization programs. We have also our generative design techniques that we use. And one of the keys to implementation is integrating all of our predictive capabilities into the project team in the most effective way. And so the way that we do this is through an integrated in silico discovery strategy. Every project and every project team has basically a mandate and expectation that there's a strategy specifically dedicated toward how we're going to use in silico tools to execute the project. And this is shown here in an example of SMARCA2. This was an oncology project, a very exciting and difficult target. We worked on SMARCA2 for quite some time, and the breakthroughs that we had in the project really came when we started to seriously apply in an integrated way an in silico discovery strategy. And the key to having an effective strategy within a project team is to know where you want to go. If anyone happened to catch Anthony Rowe's talk yesterday, he talked about the drug discovery process as really needing to know the coordinates on a map and where it is that we need to go and what are the rules of the game. So first and foremost, to put in an effective in silico discovery strategy, our project teams need to identify the key aspects that are going to make or break the project. And so in the case of SMARCA2, potency was essential. We had to improve solubility. We had to make sure that our clearance sort of matched the PK profiles. And then the second bar on here is we also had to have high selectivity between SMARCA2 and SMARCA4, which is very difficult to achieve because they're very similar proteins. But knowing where it was that we wanted to go allowed us to put in place an in silico design strategy that, as we explored different vectors on the compound, which is largely masked in this presentation, we had our objectives known and were able to put in place the right methods for enumeration, as well as for screening through our machine learning predictive filters to end up with a decent sized set of compounds that could be synthesized, could be analyzed by structure-based methods and allow us to identify highly potent SMARCA2 compounds that met all of the criteria. We didn't totally have selectivity dialed in, and so in the second sort of phase of the project, knowing everything that we had in place could continue to be applied, we added in a layer of selectivity filtering, which we had identified in a structure-based sort of signal that we could apply to compound triaging. And here, if we were to make all of the compounds that we wanted to make, which are all the red compounds, they all predicted not to be driving selectivity. And so we selected just a small number of compounds to make, which turned out to be half selective, half not selective, which really boosted the efficiency of identifying selective SMARCA2 compounds in this process. And so we can say that for a very challenging target, we didn't really make breakthrough in the program until we had an integrated in silico discovery strategy that applied all of our machine learning and AI tools to actually get the job done. And so finally, I want to talk about sort of later stage in a project when we're thinking about pharmacology and what potential dose might look like in a human. We have spent a lot of time, as I already mentioned, developing technology to allow us to predict from chemical structure what we think the human predicted dose will be. And this has had a major impact on virtually all our most recent projects, and was really a matter of distilling large volumes of data, combining that with a bit of PK data that we already had from in vivo preclinical studies as well as human studies to create a model that allows us to triage compounds and basically predict what approximate level of dose that compound will need to be effective in humans. We can then generate some PK data on compounds that we're making and further refine the model for improved accuracy. And what we found is that this actually has a dramatic impact on project teams' sort of time through cycle. So here's an example with a very difficult project called targeting MASP3. It's a protease target. And what we've been able to do in this project and many other projects is to see that there is a nice correlation between our predicted PK and our actual PK, and to use that those models to actually go straight from compound synthesis to animal experiments without running the in vitro assays in between. And that saves us plus or minus 2-3 weeks in the process. It allows us to go through the cycle much faster. And if you add that up over a number of cycles, like 20-something cycles perhaps, the time savings is enormous. And so this type of tool has readily been adopted in the process because this is the kind of thing that people want, because they want to move their project faster, they want to make decisions quicker, and it actually provides data that is projecting out into the future. So these are tools that have been developed and are being deployed over the last, say, 3 to 4 years. Where do we want to go from here? And our differentiation strategy is to really drive in 3 different areas: to do much better at driving design. We want to combine different types of data to have much better models, to be more predictive and drive design at a much higher level than we have been able to in the past. We also want to drive the make side of this cycle much more efficiently and much more successfully. So we're investing in how we do the make side of this better, both automation, intelligent automation, as I mentioned, as well as investing heavily in predicting reaction conditions for small molecule synthesis, expression, for large molecule production, and many other areas in the different modalities I mentioned. And then critical to what we see as a differentiation strategy is how we're investing in testing. Testing we don't always talk about, but testing actually is a huge part of generating much better models. So we're looking at ways in which we can really deepen and expand the richness of the tests that we do on the molecules that we generate, and then reincorporate that into better design models. Now, the reality is, and what we've heard all throughout this conference, is that yes, we all want to make better models, we want to generate more data, and we actually want to do more sophisticated training of more sophisticated models. And this is all increasing at the same time. And that leads us into a position of having this multi-pronged challenge of dealing with sort of an explosion on all fronts at the same time. We have the same level of resources, we have the same people, we have the same challenges within our own sort of pharma discovery and development environment, but our ambitions are high, and what we need to sort of corral to make the most of what the future of AI has to offer requires us to figure out a way to efficiently address a sort of exponential growth on all these different fronts. And this is just not our idea. This is happening out there in real life. A very nice slide put together by our friends at EPAM. Bryn Fenwick created this slide for me, and it shows from Epic AI that in the real world, we are increasing on all these fronts at basically an exponential level. Do we have the technology? Do we have the system and infrastructure in place? Are we planning and thinking about how to deal with this emerging explosion on all fronts? And so this is our call to action, a bit the serious part of my presentation, which is we really need to think hard about how we're going to maximize impact of AI and realize its promise in drug discovery and development, and ensure that we have the technical infrastructure and the culture in place to allow us to capture the opportunity. And I would just say that we have already realized this, but we continually need to think about it even deeper, and that is the old ways of working are not going to be sufficient to meet the demands of now and into the future. We have to be nimble. We have to work together as a team. We have to be much more fast-moving and agile. Our platforms have to be seamless at integrating new types of data. And we have to rethink how we're budgeting and paying for IT infrastructure as well as computational compute and other resources. And so I would like to present 3 different areas that we're talking about within our company and that we should all be talking about as scientists and engineers who are working at the interface of IT and wet lab data and drug discovery. And that is that we need to integrate technology within the business at a higher scale and at a higher level than we have in the past. We have to totally abandon the service model and ticket model. We have to work together as sort of cross-functional teams with transparency to the needs and challenges of the broader team. We have to break away the silos, and we have to think about what we're doing in project sort of format, just like we would with a drug discovery format. So this would address much better how we get the job done. How are we going to keep up? Platform investments, creating platforms that allow us to really rapidly and seamlessly bring in new types of data or new types of models, test them and deploy them in a rapid fashion that can actually be useful at a large scale across many different scientists in a very complex sort of environment. This is something that we really need to do a better job at, and we're really working on this within our company. And then rethink how we budget for technology. This is something that is just emerging as a topic, but technology investment has to be thought of in the same way that we think about synthesizing compounds, the way that we think about wet lab resources, the way that we think about investments in the day-to-day operations of drug discovery, and that it is a combined resource investment that we need to make. And it has to be budgeted and thought of differently so that we get the right level of resources to address the challenges and make the most of the data that we're generating. And so again, this call to action is to really go back to our teams, go back to our companies, and really think about how we can reshape and help reshape the team model, the infrastructure, and how we think about investments. Just a little bit about this in my last couple of slides, how we are striving for this within J&J. We've put together a sort of platform way of thinking about technology innovation, piloting new technology, assessing its portfolio value by really looking objectively at, does this bring value across more than one project, and then productizing that and bringing that widely to the portfolio. I mentioned the in silico discovery strategy within teams. This is an important part of portfolio-wide deployment, but actually having a workforce that understands that we need to innovate continuously. There's so much innovation happening. We need to be able to access that. We need to be able to do that ourselves, but we need to also be mature enough to bring that to a level in which it can be deployed and have usefulness across a very complex portfolio. And so this sort of working together model in a very transparent way has enabled us to have some benefit from working like this. And I just want to end my talk with an example of CHIMP. This is a chemistry inference multimodal pre-trained platform that our teams have put in place that is a true multimodal graph-based architecture that allows us to bring in not only all of our chemistry and all of our in vitro data, but to bring in quantum mechanic information, to bring in image-based, high-content imaging-based data, and eventually many more omics to the mix, and has had tremendous benefit on the performance of our models. As you can see in the lower right-hand corner of the slide, our conventional approach can lead to erosion over time as we get into, say, new chemistry space. Using a chimp-based architecture with multimodal data, we have much less erosion of our model over time as we explore new chemistry space. And so this is actually having a big benefit that I already talked about. Just a quick slide to show that normally we would have pretty dramatic erosion over time as we moved away from where we had trained and where the chemistry has gone. Using our multimodal architecture, we actually train up to 2022 data and then look at all data past, and there's virtually zero erosion over time. And I'm showing two different models here, ChILOG-D and EPSA. This is a fantastic result, and we're super excited about how this will apply to sort of our next generation property models across the spectrum. One last piece here, which is on the modality agnostic approach and leveraging across modalities. In small molecule, we want to expand and be proficient at much more complex small molecules like peptides and like targeted protein degraders. But what we found is our models that are largely based on conventional small molecule data do not perform well in more complex modality environment. But by using a chimp architecture, by using the multimodal architecture, we have shown that we actually have good prediction now transferable to peptides, shown in the left-hand side of the slide. And we are getting some performance, about half compounds are predicted in peptide space. But even more exciting, when we add in a bit of peptide data to the training, we actually increase performance dramatically for peptides, which is what you'd expect and is shown on the right-hand side. We also have an impact on our conventional small molecules as well. So this learning transfer, knowledge transfer is happening actually at both scale. And we don't exactly know the reasons why. It could be the quantum information, could be the more biological information through images. We don't actually know, but we know the performance is there. It allows us to leverage our data now more efficiently and effectively across other modalities. And with this, just one last example where we applied chimp architecture to a PROTAC project, very difficult small molecule, 2 different binding domains separated by a linker. There's a lot of iteration, a lot of ways in which we could attack sort of optimization. And through massive iteration and then property model filtering, we landed on 140 compounds that were predicted to have the right kind of PK properties that we wanted. And 39 were synthesized. These are very difficult molecules to synthesize, so pretty limited in what we can do. And found that 8 of the 39 had really breakthrough results in double digit in oral bioavailability. So these 8 compounds had double-digit oral bioavailability of a small molecule that's not normally able to be absorbed after oral administration. And this has led to a dramatic sort of a breakthrough in the program and a rapid movement forward to the clinic of a compound with phenomenal oral bioavailability. I don't think we could have achieved this without our new investment in modeling. So to end my talk, the opportunity is there's so much more we can do. We're just at the cusp of what is possible. We're seeing the benefit. We're cutting through the hype. And it's our belief that we will continue through this sort of approach to have impacts on our outcomes, including class-leading molecules, faster development and discovery timelines, and better results in the clinic and better results for patients. And with that, I'll end my talk and thank the folks from J&J who helped me put this talk together. Thank you. Yeah. Okay, so I'll be available after for questions. We have to move on.